So this topic described that the CRISPR-Cas9 is a revolutionary genome editing technology. Basically, it enables the precise modification of animal DNA. It has been widely used in the research, agriculture, and medicines, and it has been raises both the scientific opportunities and the ethical concerns. So this diagram showing the four different animals in which the CRISPR technology have been done on the basis of their model organisms and the process of their traits. So what is the CRISPR-Cas9? Basically, it is the clustered, regularly interspaced short palindromic repeats. It has been derived from the bacterial immune system. Cas9 enzymes cut the DNA at a specific locations. So it allows the insertion, deletion, or modification of the genes. So CRISPR-Cas9 is the C stand for the clustered, R stand for the regularly, I stand for the interspaced, S stand for the short, P stand for the palindromic, and R stand for the repeats. While the Cas9 is the CRISPR associated protein, which is the 9. So the mechanism of the CRISPR-Cas9 is that, for example, it guides the RNA, which we call the gRNA, firstly identifies the target DNA sequence. Now the Cas9 enzymes create the double strand breaks. So in the third step, the cell repairs the break by the non-homologous end joining and the homology directed repairs. So the CRISPR mechanisms are the four-step mechanism, and in this diagram it has been observed that the target DNA sequence is firstly identified. The second is the guided RNA, which we call the gRNA, with a complementary DNA sequence bind to the target DNA sequence. The third step is that the Cas9 enzyme use a specific DNA sequence, which we call the PAM, to find the correct spot. So it's bind to the guide RNA and cut both strands of the DNA. Once the target sequence is cutted, there are three proteins results depend on the CRISPR system programming. The first protein is the that is responsible that cut is repaired by introducing a mutations in the DNA. The second protein is responsible for some of the enzymes which are engineered to make cut on either sides of the target DNA to remove the target DNA and rejoin the ends. The third step is the DNA repair system can be hanged to insert a sections of DNA into the genome. So there is some applications of CRISPR-Cas9 in the animals, for example, for the disease modeling, for genetic improvement of the livestock, for drug testing, conservation biology, and for the veterinary research. So this diagram showing the application of CRISPRs in the animal modeling. So it has been also important for the scientific implication, for example, for disease models, such as the creation of the animal models for cancer, neurological disorders, genetic disease, improve the understanding of human disease, and accelerate the drug development. So this diagram showing the different data, understanding, insights, collections, policy, and the question. So in the animal health, it has been observed for the development of the disease resistant animal to reduce needs for the antibiotics, improve the animal welfare, and control of the inherited genetic disorders. So there are the two types of the welfares, the good animal welfare and the poor animal welfare. So for the good animal welfare, it is the normal behavior, the personality, the positive immune response, the healthy gut structure and function, the normal CNS functions, the good ability to answer the stress, the excellent health status that will advance the performance to balance the gut microbiota. The poor animal welfare, for example, for the abnormal behavior, the personality, the negative immune response, the unhealthy gut sources and the function, the unnormal CNS functions, poor ability to answer stress, awful health stresses, disadvantaged performances, and imbalanced gut microbiota. So, in the scientific implication, they have been role in the agriculture, for example, to enhance the growth rate, improve the meat and milk quality, the better food efficiency, and the increased food production. So, in the scientific implication, it may also play a role in the conservation, for example, protection of the endangered species, improve the resistance to the disease, potential restoration of the genetic diversity, and help species adapt to the climate change. So there are some risks and limitations that are associated with the scientist. For example, off-target mutations may be occur. The mucism in the edited animals may be occur. The long-term genetic effect will be unknown, and the technical and safety challenges. So in the ethical implication, the animal welfare is for the possible pain and the suffering, for developmental abnormalities, to reduce the quality of the life, and the ethical responsibility toward the animals. For example, the moral concern is that, for example, manipulation of the animal life, question about the moral rights of the animals, the human dominancy over nature, and playing God debates. The interference with the natural evolution, alteration of the genetic diversity, risk of the unintended consequences, and the ethical concerns about the unnatural changes. So there is some future perspective, for example, to improve the precision of the gene editing, the better safety control, the development of the ethical framework, and the potential benefits with the responsible use. So this is all about the CRISPR Cas9 technology. Thank you so much.